Assalamu alaikum, everyone. I hope you guys are doing well. Miss Shabnam is here, and I'm really grateful to her that she gave us time और उन्होंने हमारे लिए वक्त निकाला हमारे लिए. She's available to help us out, and I'm really grateful to her. किसी तारुफ की मोहताज नहीं है. She's one of the brightest and most amazing teachers of biology in Pakistan. So I wouldn't get in the way. Thank you so much for coming, Miss Shabnam. And
you're welcome.
I will
Thank you
leave
so
the
much.
students up to you. ठीक है. So
Thank you,
students,
sir.
you're in good hands. All the best.
All the best. Thank you so much. Assalamu alaikum. Dear students, my name is Ma'am Shabnam and my students call me Ma'am Shabnam, okay? Today I'm just going to focus on exam tips regarding your IGCSE and O-level syllabus. So first I'm going to brief you on whatever the latest syllabus changes, and then I'm going to give you proper exam tips as well. How to solve your paper, how to solve particular parts of the question, keeping in view the variable numbers and all that, keeping in view the command words and none. So first of all, let me share with you, because it's very important to first share one by one. First, O-level syllabus I'm going to share, because major changes, O-level syllabus may, whatever the changes this time, because it's all totally it's more or less similar to IGCSE syllabus. So the syllabus code for O-levels is 5090, right? And this time, this is what the revised syllabus organized into 19 main topics from 17. Number one, we have to just see that, okay, what is the major area? What is the major area here? So first of all, we can see here that first topic is classification. Classification is added here because it wasn't in the previous syllabus included. So classification include, viruses, bacteria, fungi. So viruses, bacteria, fungi, even in O-level syllabus used to be in microorganisms and biotechnology topic. Biotechnology and organisms topic. I hope you, this is what one of the syllabus classification topic. And then we have another topic is next topic, movement in and out of cells. Movement in and out of cells is basically, it used to be diffusion osmosis topic. Diffusion and osmosis topic. So this is the same, but just the topic heading, got changed. Then next topic is biological molecules. Biological molecules is basically based on nutrients topic. It used to be in nutrients topic. So nutrients may, in O-levels, you did that basics of biological molecules like all those carbohydrates, proteins, fats, and all. So now the topic is biological molecules. But in this latest syllabus changes, biological molecules, there is another nucleic acid is also added a bit. Nucleic acid is mentioned in first classification topic as well, which is DNA basically, and then biological molecules also added, and then we will be doing in inheritance also, and even we will be doing genetic modification. So nucleic acid will be repeated in most of the topics. Next topic is based on enzymes basically. So enzymes is it's basically the same, and then plant nutrition, then transport in flowering plants, human nutrition, human gas exchange. It used to be respiration based on gas exchange and respiration separately. Now it is separated as gas exchange and respiration. And then transport in humans as such. And very important, the entirely new topic is basically disease and immunity. Disease and immunity is entirely new topic. So basically, this is the topic that added this time, which includes antibiotics. Antibiotics, this topic was included in drugs topic previously in O-level syllabus. So it's a medicinal drug, and basically taking an example of typical penicillin as an antibiotic, which we extract from, Penicillium fungus from 12. And then there is malaria. Malaria they have added in this topic. It used to be in parasitism, Parasitism means the Plasmodium malarial parasite, how it causes complete life cycle. So this is also concerned with this. Then excretion, and coordination and control, which includes coordination and control is very important, because actually in this topic, coordination in plants also included recently in this O-levels topic, and homeostasis comes under major topic of coordination and control. Development of organisms and continuity of life, but sexually transmitted diseases is moved to 12. Biotechnology, genetic modification. Biotechnology, it used to be separate as microorganisms and biotechnology. Now it's biotechnology and genetic modification. Next is next topic is basically relationship of organism with one another and with the environment. This is what the topic is. It's basically related to latest impact of man on his ecosystem, other than food chains and food webs and all that. What are the topics which are basically totally no longer on the syllabus include? World food supplies, nothing much to be focused on. Malnutrition, causes of tooth decay, kidney dialysis, structure of brain, drugs, effects. I would like you to just focus on these points. Just give me 1 second, because kids are asking. I'm just putting them into case, that I've started a meeting. There are many kids asking for link. Okay, just wait. Link is the same, but I don't know. Then we have got next topic, which is not anymore included. Malnutrition, causes of tooth decay, but teeth is in the course. Kidney dialysis is no more in both IGCSE and O-levels. Structures of brain, drugs, the effects, all these are also included. Syphilis is sexually transmitted disease, which is no more included. They have taken out drugs of abuse. They have taken out all drugs of abuse. They have taken out from the syllabus. Advantages of breast milk over bottled milk, they have taken out of the syllabus. Birth control means contraceptive methods. They have been taken out of both IGCSE and O-level syllabus. Parasitism, as mentioned earlier, in terms of Plasmodium parasite, they have taken out of the syllabus. Support, movement, and locomotion, they have taken out of the syllabus. So this is what not included anymore. Now, what are the new topics for 5090? New topics include disease, cholera, and the importance of clean water, coordination and response in plants, conservation of fish stocks. So this is basically disease is included in immunity and disease topic. And cholera is also included, which was not previously included in the syllabus. Coordination and response in plants also they have added, or conservation of fish stock they have added. So this is what, according to the syllabus, O-level syllabus, which basically were topics not included and new topics added. So I would just like to now overall, I'm just giving you the topics lists, overall for 5090 O-level syllabus. It's cells, classification, movement into and out of cells, biological molecules, enzymes, plant nutrition, transport in flowering plants, human nutrition, human gas exchange, respiration, transport in humans, disease and immunity, excretion, coordination and control, coordination and response in plants, development of organisms and continuity of life, inheritance, biotechnology and genetic modification, relationships of organism with one another and the environment. So this is purely O-level 5090 syllabus. Now I'm just going to focus on IGCSE syllabus. IGCSE syllabus I'm just going to focus on. This is based on basically these are the topics. Characteristics and classification of living organisms, organisms of the organism, movement into and out of cells, biological molecules, enzymes, plant nutrition, human nutrition, transport in plants, transport in animals, diseases and immunity, gas exchange in humans, respiration, excretion in humans, coordination and response, drugs, reproduction, inheritance, variation and selection, organisms and their environment, and human influences and on ecosystem, biotechnology and genetic modification. So these are the topics which are basically now included in IGCSE syllabus. So keeping in view IGCSE syllabus, I am going to focus now on paper 4. Just give me 1 minute because I need to, ask Sir Bukhari, because kids are, calling me and asking me for the, basically, for the link. Okay, so we have actually extended theory mein IGCSE we have paper 4. So paper 4 is for IGCSE 0610 syllabus code 1 hour 15 minutes. Okay, just give me 1 second. I'm just going to just passing out on this, forwarding this link. Just give. So I'm sorry, I'm back now. Okay, so paper 4 extended 1 hour 15 minutes, and it will be of 80 marks, total 80 marks, and based on short answered and structured questions. And questions will be based on the core and supplement subject content. Other than that, this is for basically for paper 4 description, duration, total marks, and for IGCSE, and for other for 5090 O level syllabus, this is what the paper is. Paper is going to be paper 2 for IGCSE syllabus. Duration is 1 hour 45 minutes, marks 80, and there will be no section B or C for free response questions. All short answer structured questions with variable marks and variable numbers. So they will be there. So this is for syllabus of 5090 O-level syllabus. So now I'm just going to give you a little bit tips on how we are going to go about for theory paper. How we are going to go about for theory paper. So let me first, focus on tomorrow's paper. What type of questions you are going to get. I will show you the structured, some questions also, sample questions, how to start with the paper and all that. But the thing is, main, you have to base, based on your practice on how to solve the question. The main important thing is command words. Command words are very important. Command words are have been highlighted and their meaning explained. Like for example, I'm going to show you the command words like explain, describe, contrast, outline. Let me show you the command words also. So you should know that these command words are very important to be used in the questions. So when you solve the questions, you should be able to focus on questions, right? So basically, what are the command words? Now, in the exam, when you go tomorrow, inshallah, in the exam, read the instructions. Number one, important point is read the instructions carefully and answer the right number of questions from the right sections. I would like you to recommend that first you read the whole paper, and before starting your question, suppose question number 1, you read all the parts of the question and then start solving the question parts. So read the instructions carefully, answer the right number of questions from the right sections. Another very important point is that do not answer more questions than are needed. But obviously, there is no choice in your paper in IGCSE and O-level anymore. You have to do all the structured questions. All questions are compulsory. So this will not work here, will not gain your marks in the examination, but you have to do all the questions, so it's compulsory. Next part is plan your time. That time management is the most important part. plan your time according to the marks for each question. You know, for example, you have 1 mark question, you have to give 1 minute, you have 2 marks question, 2 minutes, and so on. And remember one thing, that in this type of paper, all the questions are structured questions, and all structured questions you will see that they will be basically based on variable numbers. So maximum marks of question, even you can get 6 to 7 marks, but not more than 1 part, but maximum 1 to 2 parts, you can get 7 or 6 marks based question, which can be based on a comprehension type of question, or based on some graph related question, some statistical data table related questions. So you can get all these questions, in which they ask you describe and explain. They can use more than 1 command word. So it depends on the number of lines. Suppose 7 marks question or 6 marks question, you will get 12 to 14 lines to be used. So obviously it depends. So plan your time according to the marks for each question. For example, a question worth 3 marks. If it's 3 marks, requires less time and shorter answer. 3 marks means, suppose if they ask you, it depends on the command word. If they ask you 3 features, you have to write according to 3 features. So it's 3 marks you will get on 3 features. But if they ask you something to describe, so obviously 3 points relevant points you need to write. So shorter answer than 1 word, 10 marks. Up 10 marks they have mentioned here, but you will not get 10 marks question anymore as such, because I'm going to share with you 2023 specimen papers also for IGCSE and O levels both, and even for IGCSE February March paper also I'm going to share, so that you should know how to attempt the paper as well. If a question has several parts, so it depends several parts, then the parts with more marks will need more time and more developed answer, obviously. Suppose your question carries 3 parts. So 1 part is of 2 marks, 1 is of 1 mark, but another part is for 4 to 5 marks. 4 marks are very common, marks which are mentioned in different parts of the questions. So it depends on, So next he's saying that you need to look for details. You need to look for details that indicate whatever I'm explaining right now. It's common for both O levels and IGCSE. So please focus that you have to understand that this is common for both streams of boards. So look for details that indicate how answer or the depth of answer required. For example, the question describe. This is one of the very important command word or term to be used in the question. Describe in terms of the movement and energies of the water molecules, how evaporation takes place, is allocated 2 marks on a paper. This shows that you must make 2 valid points and you must refer to movement and energy of molecules. So wording such as some molecules have more energy than others, and these leave the surface will gain both marks. So that it depends. This is very important to be noted. This is very important to be noted, right? And then you have got another very important point is, next point, do not leave out questions. Do not leave out questions or parts of questions. Remember, no answer means no mark. What he's saying is that it's better to, instead of leaving any part of the question blank, even if you're not sure about the question and you leave it blank, you will get 0. Obviously, it will be unclassified. So that's why he's saying, okay, do not leave the question, better to attempt some, of that particular part of the question. Next, he's saying, read each question very carefully. You need to read each question very carefully. So it's very important to read each question very carefully. Number 1, identify the command words. Already, shared with you that command words or terms are very important used in the question. You could underline or highlight them. Very important thing is that when you get your question paper in front of, question paper is yours. Don't worry about that if you are going to highlight or underline within the question what is he asking for. So it will be dirty or whatever. You must highlight or underline whatever mentioned in the question, the command words, what he is asking for. So you can underline or highlight them. Number 2, identify the other keywords. Keywords are very important. Keywords based on each particular topic keywords they give before the topic, and perhaps underline them too as well. Keywords or key terms to be used, biological terms, he's saying. Number 3 is saying, try to put the question into your own words to understand what is really asking. Yes, you can put the question into your own words if your expression is very good. Otherwise, I know that you were supposed to learn by heart, very important, all the, questions, so that you were supposed to answer the question by learning the same standard answers. So better to reproduce whatever you learned, but it depends. Suppose you have learned a very, extensive type of elaborated answer, and they have squeezed the question in terms of command words, and the marks are less, so then you need to squeeze it. But if you have content, you learned a content which was less, and they have asked elaborated one, then you can little bit elaborate it in your own words. Next point, what he's saying is, you have to focus on read all parts of question before starting your answer. Already I told you in the beginning that you have to read all the parts of a particular question. Suppose question number 1, you read first part in which he says describe Benedict test, and you start writing the answer in terms of excitement without reading next part in which he says explain separately. So better to read all the parts, so the parts are connected basically. So you need to know how much you need to write in each respective part. Think carefully about what is needed for each part. You will not need to repeat material. There are no marks for repeating any material. Suppose they ask you describe, means you need to describe the mechanism or principle, in terms of description. But if we ask you explain, you have to give the reason. Whatever the reason, you have to give explanation in which you must have learned because. Because means it's the reason basically he's asking for. Next point, he's saying, look very carefully at the resource material you are given. Read the title. Read the title, key, axis of the graphs to find out exactly what is showing you. You get different type of questions. You can get question which is purely based on direct relevant, questions. So it's easy to answer those questions. Second, you can get question in which you have a big comprehension, just like in English, and they provide you with the information in that particular comprehension. So you have to read and focus on comprehension, underline whatever needed information provided and whatever needed to answer the question, and then read the question and use them in your answers. Second, you can get questions based on statistical data, tables, charts, graphs, very important. Sometimes they say describe a graph, explain a graph. In tables, statistical data, maps are given. So you have to use the quoted figures. Quoted figures are very important. You have to see whatever the trend going on in the particular graph or the diagram or flow chart and all, and you have to pick information out of it. So you have to mention, suppose graph of limiting factor in photosynthesis, or graph of limiting factor in enzymes, or graphs of enzymes. You have to see that what is the trend going on, or graphs on population size and whatever. So you have to see that. You have to quote the figure, even transpiration rate and all that, different type of graphs are given. Next, look for dates, scale, and location. Try using colored pencils or pens to pick out anything that the question asks you about. Colored pencil, this is he's saying in terms of when you are basically revising something. Now, answer the question. This is very important. Use your knowledge and understanding. Do not just write all you know. Only write what is needed to answer the question. I know that we as teacher, we provide you with maximum information needed. So you have, based on each topic, you have maximum information needed. But you have to see whatever in the question, whatever he's asking, examiner is asking for. Do not just mess with the whatever, reproduce your answer and reproduce the diagrams and those ones. You have to see that. This is very important. Next point is plan your answers. How you are going to plan your answer tomorrow, inshallah, for paper paper 2 for O levels and paper 4 for IGCSE. Both of them are theory papers. Theory is basically needed. So clear, concise, well ordered, well augured, well supported answers get marks then along. Well ordered means, sometimes you have a long answer. For example, they ask you, what are the factors responsible for the transport of water through the xylem vessel? So you have to see the logical sequence of events going on. For example, insulin production in genetic engineering, you have to go on the sequence of events, how you first, identify the required allele, and then you isolate the allele, then you remove the allele using different enzyme, and then you, insert it into the plasmid bacteria. So sequence of events are very important. So, so that repetitions answers you, and quality of the answer is very important. Instead of repeating, elaborating the points, you will not get marks at all. Okay, you're going to waste your time, you're going to waste your energy, and examiner is not going to be impressed that you have written such an elaborated answer in O levels and A levels. Next point is, use the resource material given in the question to support your answer. One thing is very important. You have whatever syllabus they provide you in the beginning. So in the syllabus, they elaborate, each and everything, but still around 30 to 35% syllabus is basically unseen information. Unseen information means which is not included in the syllabus. So anything which is not included in the syllabus, they provide you information in the form of, comprehension, whatever they mention. So you have to see that whatever information provided in the comprehension or in the question. So you can see to it. Scientific terms in your answer, very important as much as possible. Obviously, scientific terms mean, since it's biology, so use maximum biological terms. Biological terms instead of writing in your own answer. For example, if I say substrate is complementary to the enzyme, so complementary word is very important. Bases in DNA, adenine is complementary to thiamine, guanine to cytosine. Same is the case with active site. Get saturated or occupied, or for example, for, enzymes. So for key terms to be used in exosmosis, endosmosis, hypotonic, isotonic, hypertonic. So depending on those key terms are very important. So you need to use them as much as possible in your material. Then make sure you are confident with your calculator. So calculator, you can take your calculator, obviously. You can take your calculator, particularly using powers of 10. Calculator is important to take along, because in both IGCSE and O-level theory paper, as well as ATP, even MCQs, you need a calculator. For theory papers also, sometimes they ask you to, calculate the percentage increase or percentage rise in, between two different values given in the graph. Percentage means you have to, initial, difference from the final, and then divided by initial value into 100. So this is how they go about percentage change in length, in potato tubers and stems and all that, or, different types of, you have to see that. So for this, you can use your calculator. Same is the case, magnification, for ATP paper and all. So there are so much to be used. You can use those as well. Okay. Then always show you working in calculation. Very important thing, very important. Most of the time they ask you, show you working in calculations, so that you can gain marks for your method, even if you make a mistake with the final answer. Very important. This is very common mistake. Kids, they always, they do it. Frankly speaking, telling you, one thing is, if we ask you, suppose to calculate the percentage rise, percentage rise in population or whatever between two values, or percentage change in length or mass in case of potato tubers or whatever. So for you, or magnification calculation, you must write. There is a paper I'm going to show you, 2000, basically, paper latest 2023, in which biology and chemistry, when there are questions which are common for ATP and for ATP as well as for theory paper. So there is a question on germinating, pollen grain get germinated, and when you immerse it into different sucrose solutions, so what happens? So there is a question on calculation part as well. So when you do the calculation, you do your proper calculation, you do a proper work for calculation to show the steps, and then you write the answer. So in this case, if your answer is even wrong due to whatever decimal points you have not mentioned, or whatever round you have not rounded off the figure, whatever, or because they get an impression that you have cheated in terms of the answer, but you don't know exactly what is happening and how to do the calculation. So must do your rough work. Same is the case with inheritance when you do the Mendelian process. When you do the Mendelian process, please, you can do the genetic diagrams, you can draw the genetic diagrams, write always, you have margins. Don't write, I don't want, don't write, obviously, just write within, leaving out the margins. But you can use for rough works little bit space and show your rough work for inheritance, Mendelian crosses as well. Make your key and make sure that there is a Punnett square or checkerboard or whatever, or genetic crosses. So wherever there is a rough work, always use a rough work. Even you can use pencil for that. Even you don't, no need to erase it. No need to erase it. But if your answer is wrong, in terms of pen you have written, so just put, you know that you cannot put a blanco or erase it. So you put a small, line, and then you rewrite your answer either above a line or below a line. So it depends how much answer you need to rewrite. Is it only one word you need to rewrite, or is it the whole answer you need to rewrite? That's very important. So that's what he's saying. So you have to be very careful about these things. Okay, so always show your working in calculation so that you can gain marks for your method. Because if suppose if you do something wrong, they will give you marks according to your calculation and your answer. It depends. And always include units. Now units are very important. I will come to ATP later, ATP paper, but units are everywhere. Whenever, suppose you have a graph question given, you have to describe and explain. So units are very important. For example, units, there's a table given of independent variable and dependent variable, and you have different type of units mentioned. So you have to see that temperature units kya hai, molar concentration units, whatever. So units are very important. Even in magnification, you know that millimeter may we take, usually in biology, we take millimeters may, instead of taking centimeter, millimeter may we take the lengths and all that. So that's very important for, to be taken. So other than that, units are very important. That's what he's saying. And next is, vague answers. I hope you understand what is meant by basically, vague descriptions. Try to write clearly and concisely using the correct physics terms. Correct physics terms, means that you have to write the correct terms to be used. You have to, for example, whatever the terminology for in terms of physics or chemistry. Suppose 3 marks question you can get on balanced equation. Very important, very common. Photosynthesis, aerobic respiration. Anaerobic respiration, they can ask you to write a balanced equation. So 1 mark for reactant and products, 1 mark for writing the correct formula, 1 mark for balancing an equation. So you have to be very careful about this as well. Use a sharp pencil for graph work, taking care of plot. Sometimes you get a grid and you have to draw a line, graph line. You can use a pencil for that. And how to draw a graph in ATP also, but here also, if you need to draw a graph, you know that how to, either you put a cross or you put a dot and circle around if you need to draw any line on the grid. So, or you can, they can ask you to calculate any, any unknown concentration or water potential. I hope you know what is water potential in osmosis topic, with a small neat cross or to draw a thin best fit line. So it depends on what type of line they ask you for. If it's a line graph, depending on independent variable, which is taken on x-axis, dependent on y-axis. Independent variable is basically the one which varies. It is under experimental control. Dependent is the one which based on observation results. And this is all, even this is common in theory paper as well. So you have to see all this about. At the end of calculation, ask yourself, is this answer sensible? Suppose there is a graph and they ask you to find unknown water potential, or they ask you to find out the unknown concentration of enzyme, whatever. So you have to see this all. Make sure you answer the question set. You will gain no marks for merely repeating the facts given in the question, as already mentioned. Make sure your writing is clear. Writing is very important, readable writing. So it's better, let me just complete this, then I'm just going to, it's better to write the, readable writing should be very clear. It should be readable, or it should not be very big, it should not be very small. It depends that it should be very clear. For example, I'm giving you example. In inheritance Mendelian process, you have to, sometimes they give you their own key to give you the alphabets for dominant and recessive allele. And if they don't give you alphabets, you have to choose your alphabets. So what is basically easy to choose the alphabets? If suppose I'm writing here, if capital S, capital S and small s, you can see here, capital D and small d, right? Capital A and small a, right? Capital Y and small y. So you can see here that D is a better choice, A is a better choice instead of Y, because Y, when you write for dominant recessive, most of the time, students, they don't write clearly difference between the recessive allele with the small case, lowercase letter, or the dominant one. So that's what he's asking you to do. That's very important. Very important in terms of, alphabets given. So make sure your writing is clear, easy to read. It's no good writing a brilliant answer if the examiner cannot read it. At least I'm not asking you to write very beautiful handwriting, but I'm just saying write readable writing. Now come to the paper 4. Paper 4 advice. Paper 3 is for, paper 3 is, is different for, this is for core, but we are doing, since IGCSE extended, or paper 2 advice is the same, which I'm just, reading out these points. Number one, but again, because this is all repetition, which I've already mentioned you, the number of marks for each question or question part often gives you a clue about how many separate points you need to make your answer. Now, very important. Suppose they ask you, list the features of dicotyledonous leaf, which help to absorb maximum light. So you have to write one word answer, or they can ask you state the features. So you have to write one line answer. Or they can ask you, it depends on, list the features of prokaryote kingdom, so classification. So you have to see, is it lift, is it state, or you have to focus on whatever the command word he's asking for, and how many points, you can give in terms of marks. Suppose 3 marks question, he can, how the water moves from root hair, from soil to root hair, or from root hair to xylem vessel. So how many marks and what is the command word? So write according to the marks given. That's very important. And then next he's saying, structured questions contain structured question. Now in 5090 O-level syllabus, no more free response question. So both IGCSE and O-level syllabus 5090, they both have only structured questions. Structured questions are the one basically with the thinking approach. They are based on variable parts of each question with the variable marks. So you have to see structured questions contain many parts. Often later parts can depend on the answer to earlier parts. Now, most of the, many students, they ask, ma'am, suppose there is a 5 marks question. It's many marks question, 5 or 6 marks. And they leave 12 to 14 lines or whatever, 10 lines or whatever. And if we finish our answer, handwriting is, small, and we finish our answer according to the demand of the question based on the command words. So is it okay if we leave a few lines? So I always say, yes, you can do that. It depends. Suppose you have finished with all the required needed answer within the, instead of 10 lines given for 5 marks or 12 lines, and you have finished in 8 lines. So it's fine. That's not an issue. But if it's 10 lines given and you have just written, you have occupied only 4 lines, that's not, that's not correct at all. It means that you have missed some of the part. Examiner, he has given you something, so he's expecting you to write according to the marks and the, question given. That's very important. So basically, you have to structured parts may command words, number of marks, and number of lines is very important. How much to write? Is it the flow chart? Is it the 1 mark question? Is it the list? Is it the state? Is it describe, explain, compare, outline, contrast? What type of command word given, write accordingly. Next, what he's saying, answer the question being asked. For example, if the question asks you to name 3 other cell structures, 3 other cell structures, do not write down the cell structures which are given in the question. Obviously, you have to see that. Suppose he gives you plant and animal cell, and he labels plant may cell wall or whatever 2 structures, and he asks you other than those 2 structures. Same is the case bacteria as a prokaryote. So you have to see, you are not going to repeat whatever given. You have to write the structures which he is asking for. That's very important. That's very important. It seems to be so easy. Ma'am is telling us the same, thing, but most of the time kids do this, mistake. Know the biological terms used in question. Biological terms are very important. If I start from biological terms, if I start from, first topic for IGCSE, characteristics and classification. Characteristic of living organisms is common for both O levels and O. So if I start from those characteristics, so you have to, what type of biological terms to be used. Then the next topic comes cell organization is the topic for IG, and cell is for O level, for O levels. So you have to see so many biological terms to be used. You have to focus on all biological terms used. Then next topic is movement in and out of cell, in which the diffusion, then osmosis, then active transport. So biological terms, without biological terms, using water potential. Suppose I'm giving you example. Diffusion is very simple, whatever the definition, movement of particle from region of high concentration to region of low concentration, that is down the concentration gradient. But when osmosis definition, so we, latest updated definition is latest first I'm covering net movement of water molecules from the region of higher water potential. So water potential is a very important biological term to be used towards the lower water potential through a partial permeable membrane. That is also down the osmotic gradient. So you have to use, another thing is when you immerse animal or plant cell into different concentrated solution. For example, hypotonic, hypertonic, isotonic. What is meant by these biological terms? What is meant by plasmolysis? What is meant by flaccidity? What is meant by turgidity? What is osmosis, exosmosis, endosmosis? Different, movement in and out may, you have to active transport, very important. Active transport is very important. O levels kids also, please, you have to focus on. Active transport is, added in your as in movement in and out. So active transport may movement of molecules from the region of low concentration to the region of high concentration, and it is always obviously that partial membrane is obviously involved, but you don't need to mention always that through a partial permeable membrane. It is involved because it is active transport is only possible in living cell with the expenditure of energy, because it's against the concentration gradient. But in active transport, proteins are also involved. Those proteins are called, carrier proteins. So carrier proteins concept was already given in IGCSE syllabus. But now I'm telling you that O levels should also know that there is involvement of carrier proteins. Carrier proteins like proteins which can bring sodium ions. Suppose they have complementary shape towards the sodium ions or potassium ions or polar molecules like glucose or amino acids. So they can bring into from lower concentration to higher, in or out. So that it depends. So biological terms are very important. Obviously, if you don't, then next topic, by movement in and out, biological molecules, very important. All biological terms are the most important biological terms. You know that biological may, if I say biological molecules are 2 types of molecules, organic and inorganic. If I say organic, I hope you must have done, in chemistry also. Organic, basically, biological molecules, organic means the one which we get carbon, hydrogen containing compounds, which we get from living source. So carbohydrates, fats, lipids, which are fats and oils, and proteins. Other than these organic molecules, nucleic acids, nucleic acids. Nucleic acids is very important term I'm using here. Nucleic acid is basically, this is also a biological organic, molecule that I'm using a term, which is basically DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So nucleic acid is also, term we can use for these biological molecules. Then inorganic term we can use for water and mineral ions. Water is now included in biological molecules in both the syllabus, but mineral ions, it's an animal nutrition, human nutrition and plant nutrition. So this is how that, very important, these biological terms which, need to mention. If I keep telling you the topics, for example, plant nutrition, I use a term autotrophic. You must have heard about autotrophic. Autotrophic means self-synthesizing. Heterotrophic, directly independent on others. I am using a term, adaptive features, in terms of adaptive features. Xerophytes, the one which grow in dry region. Mesophytes, the one grows in purely within the water. Mesophytes, one above the ground, water and below the ground. So depending on these biological terms are very, very important. So we have to, same is case with enzymes, plant nutrition. Then you come to that, human nutrition. All biological terms are very important for human nutrition. Also, you should know all the, terms of enzymes, particular names, the juices, saliva, gastric juice, whatever. So biological terms where to be used are very important. So you need to, to make your expression good, you have to use the biological terms. Keep an eye on the time management. Most of the time students ask how to manage the time. The thing is, you have seen that in IGCSE, it's 1 hour and 15 minutes. Now, please, let me clarify. 1 hour and 15 minutes of IGCSE paper is according to 1 hour and 15 minutes. But why the 5090, they have 1 hour and 45 minutes, still paper? Because it used to be, old syllabus. It used to be same 1 hour 45 minutes, but there were 4 to 5 structured questions in section A, which were compulsory. And then section B and C may 2 questions compulsory, free response, and 1 either or in C section. It means that they are basically giving you the same type of descriptive questions. They are basically demanding the same answer from you in 1 hour and 45 minutes. Why you have more time as compared to IGCSE, 30 minutes more? Because you will be getting 30 minutes more descriptive question ke parts. So means that you have to see the time management accordingly. IGCSE will get a less number of parts in the particular question. Maybe they get 5 total questions or 6 questions, depending on the length of question, but it depends. So you have to be very careful about it. Time management, you have to see that. Or then you have to, after going through the, you solve one question paper, you solve first question, try to attempt all the question. If you don't know any part, please don't waste time. You jump to the next question, start solving those questions. You can get an idea before coming back. So you keep solving the question and then come back to the previous parts which you left. Suppose if you leave in any of the question. So time management according to, first you read the whole paper and spend 5 to 6 minutes, doesn't matter, it's not a wastage of time. Even if you go through the paper for more than 5 to 6 minutes, it's not wasted. Even 10 minutes, less than 10 minutes, 6 minutes you can take to read the whole paper. And then you start tick mark the question which are in your total grip. I would prefer to start from the question number 1, go in sequence, but you can leave the parts if you feel, instead of thinking and spending time, because you have to see the number of marks given. So marks, you have to lead towards the time. So 2 marks means 2 minutes, 2 maximum. You can take less, but 2 maximum. 3 means 3 minutes. So you have to see that. So this is called time management. You cannot just ignore time management. Otherwise, you can, biology is always, there's a complaint that biology is very lengthy paper. So obviously you need to write much answers, though it's not that lengthy paper. If you have done a good, grasping and, you have gone through your revisions content very well. So that's what he's saying. So time management. So that's very important. This is all that, questions depending on, theory paper, depending on how to go about your theory paper. Now same goes for O levels paper as well. Now there is a paper, I'm opening a paper, a specimen paper. Let's do both the papers. First I'm opening this O level specimen paper, right? O level specimen paper, which is basically they are going to test you on for 2023 syllabus, right? This is the paper for 2023. I know, I hope so that you all have solved this paper. I'm taking this paper as an example because I'm also keeping an eye. I'm also keeping an eye on time management because I have to cover most of the content which is needed here. Now this is paper theory paper 2 to be tested for examination 2023. Even if you have covered it, doesn't matter. But what he's saying, I would like to read the instructions also. Read the instruction which students keep asking, even they are asking right now as well. Number 1, answer all questions. Yes, answer all questions. I will read out for IGCSE also. So both are common. This is common in both cases. Answer all questions. You have to now answer. No free response, no choice. Fine. Number 2, use a black or dark blue pen. Yes, this is also common instruction to be followed. You may use HB pencil for any diagrams or graphs. I have got even, my students know I'm talking about. I have got even today also this, query that do we have to draw diagram in pencil or graph? Yes. You draw your diagram if they ask you to draw a diagram or a part of the diagram, or even in theory paper. I'm talking about tomorrow theory paper. ATP mein to obviously you have to draw, or you need to draw a graph pe grid paper pe you need to draw a line in the graph paper. You use a pencil. But if you need to label it, so you can label it in pencil or you can label it in pen, it depends. So if you label it in pencil, it's fine. Instead of label in pen, if you do something wrong, then you have to cross it and it's difficult for you to, right? Then next is write your name, center number, candidate number in the boxes. Very important. Someone was telling me that, but I saw in the, I saw on social media, Facebook, that, someone got some wrong code paper, invigilator gave a wrong code number paper. Suppose if I say IGCSE you have registered for, and you will get 5090 paper. So you have registered for. So please be careful. You have to see that are you opting for IGCSE 0610 syllabus extended or core. So you have to see extended at core. You have to see which syllabus. Number 2, are you taking O-level paper? So you have to see 5090 paper 2. You have to see that. So first of all, make sure that you have to check the paper. Done. Okay, done. Then you have got a correct paper. So you are not going to blame anyone. I am very surprised that someone was saying that invigilator kept saying, no, this is your paper and it was wrong paper. It's impossible. You stand up, you take a stand, you stand up and you just move out. You say, okay, no, this is not a correct paper. I have to give this paper. Please, you have to see that. You can complain to some other invigilator or, there is some head of the British Council or whoever. So don't do this. This is very important. Next, he's saying, write your answers to each question in the space provided. You cannot write, there are margins. You cannot write anything out of the, space provided. If you write within the margins, there will be a scanning. So you will not, your answer which needs to be included, it will be lost, So better to, instead of, don't write anything in the margins, within the margins. If you write anything which is rough, then fine. But if you need to, your answer needs to be included. So do not do like this. Can you write, so write within the space provided. Make sure that if suppose your answer is wrong, you cannot blanco it, you cannot use eraser in ink. So you just put a small line, one line. Do not redraw line again once, then second time, because the students do like this. And then you write above the answer or below the answer. It depends how much you need to rewrite. Do not use, obviously, erasable pen or correction fluid. You cannot use blanco stuff and all that. Do not write on any barcodes. As I said, do not write on any barcodes. You may use a calculator. Obviously, you may use a calculator. Better to use a calculator. Even if you are very good at your mind, mind map. Even if you are very good at mind map, do not use this. This is very important. Okay. So I hope you have to just see this. Uske baad next is you should show all your working and use unit. Obviously, Abhi, I already told you that you have to show your working for any calculation based question, and you have to mention the units, in which units is you are basically calculating. And if they give you the unit, for example, percentage is written, you just put the answer value. but they give you the unit. So it depends, right? So total marks, how many marks for this paper? For 5090, 80 marks. 80 marks, abhi I told you, 80 marks are common for IGCSE and for both. The number of marks for each question or part questions is shown in the brackets. This is also common for both of you. So whatever, remember one thing. I'm just discussing jaldi se these two papers, these typical IGCSE, one specimen paper and one for O levels. But the thing is that both are common. These papers are common because now IGCSE type topics are included in O levels as well. So there is no difference between the two as such. Now this is question number 1. You can see here. This is question number 1. Digestion. You can see here. Digestion in human digestive system is carried out by action of enzymes. So you can see here now, bring it to your mind. First question is basically, jaldi se focus on human nutrition, digestive system topic. But if you focus on digestive system topic, so you have to see that. Now, digestion, human digestive system, and he's talking about enzymes as well. So you have to see the type of enzyme. Now, since we are doing enzymes, I hope you understand when you did enzyme topic. Enzyme catalyze 2 types of reaction. They are biological catalysts. They catalyze both type of reaction. One is called hydrolysis, another one is called condensation. I hope you understand. So both type of them. If you don't know what is meant by hydrolysis, in the presence of water, complex molecules can be broken down into simple nutrients, right? And condensation is complex molecule can be formed from the simple nutrients with the removal of water molecule. So you have to see all this. This is very important action of enzyme. So the diagram is given. I'm not going to go through each part because there is no much time to cover everything. But still, this is what he's saying. Figure 1.1 jo hai done for you. Name of enzyme, pepsin suppose given. Pepsin is given. And then where enzyme acts, stomach is given. Protein is a substrate. Amino acids jo hai, they are a product. So they have filled up with this. Second, they have mentioned starch is a substrate, and name of enzyme you need to where enzyme acts, and the substrate is mentioned, which is starch, and end product maltose is mentioned. So you have to bring it to your mind. Digestion of carbohydrates. Jaldi se apne mind make digestion of carbohydrates. Carbohydrate digestion starts from mouth. Why it starts from mouth? Saliva contains, that. What saliva contains? Amylase, saliva amylase, pH is suitable and whatever. So bring it to your mind, starch convert into maltose in the mouth. Or there is no action in the stomach, and then maltose reaches duodenum, the first part of small intestine. Duodenum, how secretions coming into duodenum same time. Pancreatic juices coming containing maltase. All the most of the enzyme, can they act upon. Even bile is coming at the same time, which is not containing any enzyme. Or pancreatic enzymes, intestinal enzymes. So you have to see that. You have to fill up with this. Next, you have substrate. Enzyme is lipase, where enzyme acts, substrate, and products. Now bring it to your mind 2 things. Lipase is an enzyme which acts on lipids. So before lipase is act upon, there should be a proper process of emulsification. How fat globules can be emulsified into fat droplets. That's important. Fat droplets, they are acted upon by lipases enzymes to increase the surface area. So lipases are present in the pancreatic juice, and emulsification is done by the bile. So you have to see so many topics are interconnected in this. Amino acids are the end product of protein digestion. Now, very important. Now, what is the command word here? There are 4 marks. This is a big question. Above was very simple, 1 mark for 1 each. But here there are 4 marks, and there are more than 8 lines. More than 8 lines. So maximum 8, but more than 8 lines mentioned here. Now, describe is the command word. Describe what happens to these amino acids after digestion until they reach the liver. Until they reach the liver. So they are connecting you to so much thing. Bring it to your mind the steps of digestion. If you remember, ingestion, digestion, mechanical, and then chemical. Remember? Then absorption, assimilation, and then ejection. So you have to see how it relates. What is assimilation? When it reaches liver. So when it reaches liver, amino acids, that they cannot be stored. So how the toxic part of the amino acid removed in deamination process and all that. So you have to connect it with this. So I'm just going to show you the obviously mark scheme jaldi se, but right now just see the question. Concentrate on the question which was described for POMA. Now this is a question. This is a question for figure 2.1 shows the effect on crop yield amount harvested of using nitrogen containing fertilizers. Nitrogen containing fertilizers. I hope this is a question also very important. Now this is a graph given. Now this is very good typical question, graph based question. Most of the kids they ask. Now look at the graph based question, figure 2.1. Mass of fertilizer is given but along with the unit. Can you see here mass of fertilizer and the unit in kilogram per 10,000 meters square, whatever is given, and then crop yield also given, with unit, and the values are given, and this is the line, and you can see how it goes up from where, not from the zero starting, and then it levels off here. So if I use the word levels off or whatever, you have to see this all. Now, what is the question here? Using the information. Now, this is very common question. Using the information in figure 2.1, Now, underline in your question paper. Using the information in figure 2.1, what is the command word used here? Describe. Describe the effect on crop yield of using and increasing mass of fertilizer. Now, this is for 3 marks. So please, you have to use the quoted figures. I'm going to discuss the answer, showing you the mark scheme as well, but you have to quote the figures. And it's very easy. Only thing is, you should have an idea that you have to quote the figures and matching it with. Now, next part. Nitrogen in the fertilizer is in the form of nitrate ion. Concentrate on nitrate ion. Now, bring it to your nitrate ion. Why nitrate ion contain nitrogen? You know what is nitrogen? So bring it to your mind. Nitrogen. Why you need nitrogen? Why you need nitrogen? Very important. Nitrogen is needed for what? This is also important for one of the essential element in proteins. Now, what is the command word here? Describe how the nitrogen in the fertilizer is absorbed by the crop plants, and used to give an increased crop yield. Think of it. Nitrogen in the fertilizer is absorbed. What is fertilizer? You must have done nitrogen cycle, if you remember. The role of nitrogen cycle means the role of fertilizers, the role of bacteria in the nitrogen cycle, addition of nitrates, removal of nitrates. So, bring it to your mind. You have seen here, so what are the topics which are related in this question? So next part is, this is a different command word, explain. Explain means asking for reason. Why a farmer may decide to use a mass of fertilizer per 10,000 meters square, which is less than that needed for a maximum crop yield. Why he uses 10,000, which is less than that needed for a maximum crop yield. So you have to give reason for it. Okay? So I'm just going to discuss. I'm not right now telling you the answer. Discuss the mark scheme as well. Next part is another different state, command word. State. One type. One. You cannot write 2 or 3 or whatever. You cross 1, you write 1 only. One type of mineral ion other than nitrate. Don't then repeat nitrate. That is required by a plant and explain its importance. Suppose mineral ion other than nitrate. Is it magnesium? Is it, phosphorus? Whatever. I'm just giving you an idea. So write the importance as well. But it should not be nitrate. So it's for 2 marks. This is the question number 2 then. Now this is a question. Next question. This is a very good question. Question number 3. Why good question? Because you can see here that information provided here is basically DNA. Now, what is DNA? DNA, Abhi, I told you. What is DNA? It's a nucleic acid. Don't forget, I'm using a word, nucleic acid. Nucleic acid is a term I'm using for DNA and RNA. And what is basically, DNA stands for? You should know what a DNA stands for. Deoxyribonucleic acid, right? Nucleic acid, nucleic acid. Nucleic acid stand for, we are talking about basically here, DNA. What is DNA stands for? Deoxynucleic acid. Is that okay? Deoxyribonucleic acid. So this is what DNA stands for. Number 2, there is RNA, ribonucleic acid. RNA, which stands for ribonucleic acid. So in this, he's just focusing on basically DNA. And he's providing with information that DNA controls cell function. It controls cell function by controlling the production of proteins. Now, if you remember, let's start from the cell topic. Cell contains nucleus. You should know what type of cells. All cells contain nucleus. No. Prokaryotic cell, which is a typical bacteria, it lacks typical nucleus. There is only circular DNA lying in the cytoplasm. There is no nuclear envelope around it. But all eukaryotes mean all unicellular and multicellular organisms other than bacteria, which is prokaryote, they contain nucleus. So nucleus contains, chromatin network, which is made up of a thread-like structure called chromosomes. Chromosomes are made up of DNA, DNA carries hereditary information. It carries hereditary information for the formation of amino acids. Amino acids make proteins, right? So this is what the information he has given you here, right? And then what he's saying is these proteins determine phenotype. Now, very important term, another term here. What is phenotype? Bring it to your mind. Inheritance. Genetic terms. Hurry up. Bring it to your mind. We can start from, gene. Small sequence of DNA is called gene. Then alternative form of gene is called allele. Allele can be dominant, which can be designated by capital letter. Allele can be recessive, which can be designated by lowercase letter. You must have done Mendelian crosses, how he did experiment on pea plants. And depending on the character or trait, suppose height for tallness or shortness and all that, round, wrinkled, and so over. And same case in the animals, black coated, animal, or brown coated, or whatsoever. So phenotype, and then alleles, dominant, recessive, homozygous, both alleles same. Heterozygous, both alleles different. Dominance expressed in two forms: homozygous dominant, heterozygous dominant, but recessive only express itself in the presence of same alleles, which are always homozygous. What is phenotype? Observable characteristics. Observable, the one which can be expressed traits or observable, is called phenotype. Phenotype is basically controlled by genotype as well as environment. What is genotype? The genetic makeup. Suppose if I this is just a ab initio, it's coming the crosses also, but I'm just saying, suppose I'm talking about like tall plant. Suppose I'm writing here tall plant is a phenotype, pea plant, and it's if I write a genotype, I write like this. It can be this or it can be this, homo or hetero both, and it is basically crossed with short or dwarf. So short pea plant, short is always it's always recessive. I can write short with a small case letter of the dominant allele. So writing phenotype, parental phenotypes in terms of words is tall and short is phenotype. But when I write genotype like this, homozygous dominant, heterozygous dominant, or recessive short. So these are called genotypes. So you should know the difference between phenotype and genotype. So, so I've repeated for you this also. Okay, what is the comparison between these two? The describe now. Question is totally different, which I just focused on. Describe the structure of DNA. Describe the structure of DNA. I will show you mark scheme obviously, but I'm just repeating here. Describe the structure of DNA में you should know कि nucleic acids का we always use a polymer. Polymer का मतलब है long chain. जैसे polynucleotide. Nucleotide. Polynucleotide means long chain, just like polypeptide in protein, polysaccharide in carbohydrates, right? So polynucleotide is a long chain of nucleic acid. This is called polymer. What is the single unit monomer? It's nucleotide. It's nucleotide. Nucleotide means it consists of basically what it consists of. Nucleotide consists of three components. Actually, it consists of phosphate, which is designated by circle. It consists of sugar, 5 carbon. 5 carbon pentose sugar. 5 carbon, we call it pentose sugar, and it's of two types: ribose, which is present in RNA, and deoxyribose, which is present in DNA. Deoxyribose. Is that okay? And this is how two sugars are present, and then very important nitrogenous bases are present. Nitrogenous bases are also called organic bases. So there are basically 4 kinds of bases present. Adenine. Adenine is one of the base designated by capital A. It always pair with another base through hydrogen bonds. Hydrogen bonds, and there are basically 2 hydrogen bonds with thiamine, right? And then there is another base called guanine, which is it's held by 3 hydrogen bonds. It's basically not 2, 3 hydrogen bonds. So 3 hydrogen bonds. I'm writing here 3 hydrogen bonds with cytosine. Cytosine is another nitrogenous base. So this is all structure of DNA in terms of what is present. But the important thing is, I will come to the mark scheme, but DNA is called double helix. Double helix. It's a double helix structure. It basically consists of 2 strands running and tied parallel to one another. They run opposite to one another. So I'll show you the mark scheme as well, but it is called double helix structure, DNA. It was discovered by Watson and Crick model. You must have seen that kids they make model of DNA. It's very common. 1953, Watson and Crick model was put forward. So this is called double helix structure. There are double strands, and in between the strands, the 2 strands are made up of sugar phosphate, and in between the nitrogenous bases are held by hydrogen bonds. Then this is a problem. This is a problem. So this paper is basically for both of you. This is a paper. Even if I don't get managed, I will cover the for IGCSE paper also. February, March, I will go fast track. But this is common for both of you for the later syllabus. One parent of child. Now please focus on this. When you get any problem on inheritance, what you have to do? Underline, one parent of a child has a blood group. Achha, a blood group is very important. Blood group may you have to see that one simple is monohybrid inheritance you have done when single contrasting character present at a time, and how monohybrid inheritance is maintained in the first filial generation, which is a result of parental process between the 2. And then the first generation, suppose it whatever you do to bring the second generation, so you can cross between the 2 offsprings whatsoever. So blood group is basically, there are 4 blood groups, A, B, O, and AB. And these blood groups are controlled not by 1 or 2 alleles, it is controlled by 3 alleles, multiple alleles, A, B, and O. So AB allele, you must have done that. AB allele dominant over O. So we can use, you have to see that when you write it, blood groups made, you must have done, I will show you the mark scheme, that how we write it down. In blood groups, only in typical blood group, we need to write I. I is basically, it's an antigen. And above that, you have to, in subscription, you have to write whatever the, suppose it's A, allele, you have to write in the subscription, right? Just for example, I write like this is homozygous for A blood group. So homozygous form or hetero, IA, IO. So you can write this as well. So this is showing you the, how to write. Otherwise, you write simply the symbols. You don't need to show the antigen like I here. I hope you understood, but I'm obviously will show you. Now he's saying that parent of a child has blood group B. Now underline this. Blood group B, and the other parent has a blood group A. Child is blood group O. There is a very common question that how come that parent of blood group A and parent of blood group B, they can give rise to a child who is O. So there was a criminal case regarding that, and they were fighting for that case that, XYZ, he's not our child. They were complaining in the court. So they tested it for. So what you can see here is homozygous B and A. They both exist in homozygous and heterozygous both forms. So when both the parents, A and B, they are heterozygous, like for example, IBIO, IAIO, when you cross them, then there is a 25% probability or chance to bond with blood group O. This is what that's shown here. So I'll show you that mark scheme as well, but this is what, they have shown here. So this is very important. Complete the genetic diagram. Now, this is a genetic diagram. You don't need to genotype of parents. You are not supposed to write the phenotype, only genotype. So complete the genetic diagram, hetero with hetero. Suppose you have hetero with hetero. IBIO, IAIO. So you can make the gametes here. IB, okay, IB, one gamete, IO, another gamete, IA, another, one gamete, IO, another gamete. So you make and write gametes. Possible genotype of child, you can do the process, find out what are the 4 possible genotypes. Aram se. Possible blood group of child means it's basically the phenotypes he's talking. State the probability that the second child of these parents has the same sex as the first child, has the same blood group as the first child. So this is showing you with the same, the crosses. Now this is question number. This is the question number 4. This question number is basically, it's a very common question, just like in ATP, alternative to practical. You can see here, it can come in ATP paper also. Photomicrograph pollen grain with a pollen tube growing out of it. Okay, this is that pollen grain, when it matures, it develops a pollen tube which can enter to the micropyle and into the embryo sac, and how it double fertilization in plants it occurs. So they have shown here, and what he's saying is a student placed pollen grains from the same type of plant underlined in sucrose solutions. Sucrose solutions of different concentrations of a fixed amount of time. After this time, student used a microscope, examine the grains and the tubes. So jaldi se dekho, this is the table, you must have solved it. This is a table given, percentage sucrose concentration given, percentage of pollen grain germinated, mean pollen tube length is given out. Total of underline, what you need to underline, total of 12 pollen grains. 12 pollen grains were placed in 20% sucrose solution. This is what the information is. 20% with 25 wo jo hai. So 25 divided by 100 into 12, you will get the answer, whatever. But you must have solved it, and you can aram solve it using the information. This is very easy calculation. It's not that difficult. Suggest why the mean pollen tube length was calculated for each sucrose concentration. ATP may be aayega. Why we need to mean? Mean is very important to, give you the reliable result and all that. Using the information, identify the optimal best concentration of sucrose. Wo jo best concentration of sucrose hai, jahan pe maximum pollen grain germinated. You can see here, ye dekho, percentage 8 ke upar 56 germinate hue. So this is the best one. Aram se, you have to underline whatever information given, and you can use this information for this one. Germination of pollen grain requires movement of water into the pollen grain. Now, movement of water into the pollen grain, bring it to your mind the osmosis topic. I'm just, fast, because just to cover it, movement of water depends on concentration gradient, osmotic concentration gradient. What is the outside and inside difference in terms of osmotic gradient of, equilibrium of the solutions? Suppose outside is concentrated solution, so water will move out then through osmosis. If outside is dilute hypotonic solution, water will enter into the cell. So that's how the water moves through osmosis, from higher water potential to lower water potential. So suggest why placing pollen grain in solution with a higher sucrose concentration in your A3 answer may result in a lower percentage of germination. Low percentage isliye because water jo hai it stimulates, it helps in the maximum germination. Kyunki seed will germinate in the presence of what are the condition? Oxygen, optimum temperature, or moisture, water. So water is needed. So water kam ho jayega. If water will start moving out, to water kam ho jayega, to germination slows down. So describe the route. Now this is term, describe the route taken by a growing pollen tube in a plant and explain the importance of route in plant reproduction. Route taken by growing pollen tube in a plant, explain. अब इसमें 2 terms हैं, don't forget please. Describe or explain importance of this route in the plant reproduction. तो अब ये 4 marks का question है. You have seen that के maximum अभी तक we have seen that 4 marks question is given here. उसके बाद this is very good question for the comparison. Comparison में भी lot of confusion regarding students. Always see के what is the first term given here. Now see here. Compare each of the following processes. Suppose so agar first term is given aerobic respiration and second is given anaerobic respiration, so pick up the aerobic respiration key points or features and compare it with anaerobic respiration instead of picking up anaerobic once and then aerobic and then mixing it up. Use the first feature, pick up the information of first term given and compare it with the second term. Vice versa bhi aap kar sakte ho, but this is what that proper way. Uske baad, this is very common, it's not difficult at all. Next is anaerobic respiration. 3 marks ka, so you have to give 3 points ka comparison. 3 points normal, obviously. Anaerobic respiration in humans. Now, anaerobic respiration in mussels and anaerobic respiration in yeast. If you remember, alcoholic fermentation and lactic acid fermentation. So please bring it to your mind jaldi se. Uske baad next is photosynthesis or phototropism. Photosynthesis is common for O levels and IGCSE, but phototropism O levels you haven't done before. Though I always tell them when we do characteristic, how the sensitivity characteristic, how the plants respond towards a stimulus. Photo ka means a light, tropism in towards. So phototropism means shoots grow towards sunlight. So coordination, जो plant topic है gravitism और ये gravitropism or phototropism के ऊपर, this is the topic that they have added here. So you must be knowing that topic. ठीक है? So how shoots move towards the sunlight and how roots move towards the force of gravity, that is towards the soil geotropism. उसके बाद ये structures they have given reproduction. Process of reproduction produces genetically identical, more individual the same, always involves one parent. Okay, jaldi se this topic is mentioned relevant to mitosis, meiosis. There are 2 types of cell divisions, mitosis, meiosis. Mitosis occurs in body cells, which are also called somatic cells. Meiosis occurs in the gonads or reproductive organs. Mitosis leads to asexual reproduction. Meiosis leads to sexual reproduction. Meiosis leads to gamete formation. Mitosis mein number of chromosomes remain constant from parent to offspring. Meiosis may reduce to half. Mitosis में जो है same characteristic pass from parent to offspring. Meiosis में variation occurs due to crossing over between the inner chromatids. That's why we are all different. So sexual reproduction में. So this is all significance also कि how mitosis is involved. Mitosis जो it helps in asexual reproduction, like budding in yeast and hydra में budding and potato में budding and artificial vegetative propagation, cutting, marcoting, and methods in plants, vegetative propagation ones, cloning, tissue culture, those are all mentioned, but asexual reproduction also occurs in, like binary fission in bacteria and in amoeba. It's not mitosis in bacteria at all, right? So you have to see that. So involved, this is all haploid and diploid. Haploid means one set of chromosome, like gametes may we have 23 chromosome, half the number of chromosome. Diploid means 2 sets of chromosome, like each and every cell of our body contain 46 number of chromosome. Number of chromosome is fixed in each and every body. Involves only cell division by mitosis. So obviously wo kya hoga? Niche now you have to see, you have to fill this. Sexual reproduction only, asexual reproduction only, both sexual, asexual. So you have to see that it's not that difficult at all. Or next 7th one, this is the topic that they have included. Now, this is the topic they have included for O levels 5090 syllabus, which is classification. Now, classification, IGCSE ones are very familiar with the classification topic. Organisms can be classified into groups. Now, you know that what is the hierarchy, how the, it starts classification. If you just bring it to your mind, it starts from the kingdoms, like animal kingdom, plant kingdom, then the phylum it comes, then the phylum ke baad, then order, then family, class, it comes to then, genus or and species. So basically, yahan pe, he's saying ke state 2 features used to classify humans as mammals. So up you have to pehle to underline state keh rahe, 2 features ko bol raha hai, or classify humans as mammals. So you have to just mammals. So bring it to your mind, mammals as animals. So mammals ke features you can write here. Then second term is scientific. Each and every organism, they have what type of organism. If I say bacteria causes TB, tuberculosis, so I can say okay, bacteria causes, so bacteria is an organism. But if I say, tuberculosis is mycobacteria, whatever I take a biological name, so that is basically the scientific biological name. Yahan pe he's saying humans ka biological name is Homo sapiens. So this is called binomial. Binomial ka matlab splitting into 2 halves. First term indicates the genus, which is always written in the capital letter, and small letter ke sath second term indicates the species, which is written with the small letter. Sometimes species of 2, but mostly aapke level pe sapiens like either Homo is genus, sapiens is species. Or state the level of classification. Homo genus ka matlab hota hai they have structured resemblances, but they cannot interbreed to produce fertile offspring. But in case of sapiens, species ki definition you must be knowing. They are they have structured resemblances as well as morphologically. Morphology means apparently, morphologically, physiologically, they can interbreed to produce fertile offspring. I'm using a word fertile. Fertile का मतलब होता है they are able to reproduce. Sterile का मतलब sterilization is different, free of contamination. Sterile means cannot reproduce. So you can just write here, I have given you enough idea while discussing the questions. Uske baad humans may be infected of infection. This is very important. You know, very important thing is aapke O-levels ke syllabus mein they have added disease and immunity. So there was no topic like immunity or in O-level syllabus earlier, but disease ka bhi as such, diseases were included, but not as such. Drugs topic was included, but now IG and O-level both, they have a concept of drugs, what is basically disease and immunity. What is pathogen? Pathogen ka matlab hai disease causing organism, right? Disease causing organism can be bacteria, is a virus, it can be protists, जैसे plasmodium malarial parasite है, protozoan है, amoeba है, whatever. So protist can be. So humans may be infected with pathogens such as viruses. तो viruses जो है वो कह रहे state two main feature of viruses. Viruses जो है they are at the borderline of living and non-living. They are not proper organism. Why they are borderline? Because non-living, because they are inactive outside the living host. You know, they only reproduce when they get attached with the host membrane metabolically, or they don't have their own metabolism. So when they attach themselves, then they reproduce. Or why we call them living? Because they have outer protein coat along with the inner nucleic acid core. Most viruses, they have RNA as the nucleic acid core. They are called retroviruses. And bacteriophage, the virus which attacks bacteria, it contains DNA. So basically, they also have nucleic acids. So that's why we can call them they are basically the ones with the they are basically living as well. D part mein, this is called now what type of you have to see that sample of human blood. 7.1 one cell labeled X, cells of this type produces antibodies. Ab jaldi se apne mind mein lao, because antibodies where did you study antibodies? Ab immunity, antibodies are very important, which are produced immunity mein. Balki transport in mammals ka topic hai, lymphocytes. Lymphocytes, white blood cell, two types ke hote hain, phagocytes and lymphocytes. So phagocytes, they engulf bacteria. They have multi-lobe nucleus or bean-shaped nucleus, whatever, and they undergo phagocytosis with the under the action of enzymes released. Lysosomes hote and they release enzyme. But to lymphocytes, they are big nucleus and they are of two types, B and T. They, the B lymphocyte, they release actually antibodies. Antibodies, they have a complementary shape to the particular antigen, which is a foreign particle. Suppose any bacteria or virus attacks your body, to uske upar antigen hoga, which is complementary to the antibody. So for example, B lymphocytes, they, when any particular bacteria enters your body, to uske upar jo hai antigen hai complementary to the antibody, to wo phir, they start fighting against it. So they have those plasma cells as lymphocytes, as well as they have memory cells. So memory cells ka matlab hota hai, when there are only antibodies floating in the plasma, so there's a short-term effect of the fighting with the disease, like pathogen. But if there are memory cells, they produce a long-term effect. So this figure is actually called blood smear. Blood smear ka matlab hai, there are different type of cells you can see here. Other than whatever X yahan pe dekho, concentrate on, could you just concentrate on this X? Yani having a big nucleus, or plus there is a periphery pe, there is a cytoplasm, but it's edge ke upar very less cytoplasm. Then you can see this type of, they have multi-lobe type nucleus, but they have cytoplasm. This is a different type of, so there are different, and these are all different type of cells given. Outline the type of ye jo X hai, labeled defends the body against infection by a pathogen. So you have to see that. Infection causes the type of cell labeled X to produce antibodies. So antibody already मैंने आपको इतना explain कर दिया है कि how antibodies do and they have they can deal with it. 8th question is, now this is a 4 marks again question you have noticed. Explain is a command word. Concept of control by negative feedback. अच्छा आप negative feedback को जल्दी से just bring it to your mind. Negative feedback we did basically in homeostasis topic. There are certain factors or, variables which in our body, features or factors which need to be within narrow limits. They need to be constant within our body. Usme very important is pH of our blood, glucose concentration in the blood needs to be, temperature, core body temperature needs to be constant. So these are the examples, carbon dioxide, whatever. So if there is any increase or decrease in any of these parameters, they should come at a steady state after such a period of time. So for example, temperature. Temperature is basically controlled by thermoreceptors. Thermoreceptor, they detect the change, fluctuation in the temperature, and then there is a negative feedback, how it increases and decreases. So they bring about the changes in the body, like what happens in hot days and cold days and all that. If glucose concentration increases in the body, you know that. So how the insulin converts glucose to help to convert glucose into glycogen in the liver. And then if the glucose is less, so how glycogen converts back into glucose under the influence of glucagon hormone. So basically, this is called negative feedback mechanism. Very important. Uske baad, isi tarah, the pH of the blood needs to be within narrow limits. There should be no increase or decrease, because each and every cell of our body ki there is a outside environment which is called tissue fluid. Tissue fluid, it's very important. All the cells, they take bath in it, and the composition needs to be within narrow limits in terms of pH and composition and all that, and temperature. Uske baad, describe how two named components of the skin are involved in regulating body temperature in hot condition. So basically, there are warm-blooded animals which are also called endotherms. You can call them, or there are ectotherms, or cold-blooded animals which are called poikilothermic. So how they maintain their body temperatures, there are so many much. Keeping in view suppose how fat insulates, how it helps to keep the core body temperature constant, how the blood vessels in terms of vasodilation, vasoconstriction, how the sebaceous gland, how the sweat glands, they help. So basically, all those structures, root hair, and how they, all these structures are important in terms of how they maintain, So this is all about the paper, which I have already, I have even, done with this all information given. So I am not going through the mark scheme, because you must have this mark scheme. Instead of going through the mark scheme, there's another paper jaldi se going through, because you must be having this paper, you must have solved this paper. Now jaldi se going at a glance, this is IGCSE IGCSE paper, the one with the now I'm doing the paper. Yes. The paper for this is for February, February March paper. February March paper 2023, abhi latest paper. Now this is the paper. Now look at this paper. Figure 1.1, simplified diagram of the circulatory system in humans. This is for IGCSE paper, as I said. So there are some alphabets they have mentioned here. Brain diya hua hai, organ, heart as an organ, lungs mentioned, liver mentioned, digestive system hai, other organs, whatever. Now what is the question? State. Now what is the command word they have used here? State the letter from figure 1.1 that represents the blood vessels that contain blood with the highest concentration of oxygen. State the letter, give 1 point, represent the hepatic portal vein. Hepatic portal vein, if you remember, you should know that the hepatic associated with liver, but whatever food comes from small intestine, it goes through hepatic hepatic portal vein into the liver. It takes the amino acids and glucose there, but the fate of the fats is different. Fatty acid and glycerol, they combine and they produce fat globules again to be distributed through lymph. So unka fate is different. State the letter represent arteries. So ye to koi itna wo nahi hai, jaldi se I'm going fast. Explain the importance of part labeled Y in the diagram. Part labeled Y. Ye dekho, explain ka matlab hai Y ko pehle gaur se dekho. Y. This is Y. So you can see here, I can see ye hai Y. I can also see that this is septum. तो आपको पता होना चाहिए what is the role of septum. Septum जो है separates the oxygenated blood from deoxygenated blood. So ये he is what is asking for the importance. So जल्दी से this is what must be written here. You have to elaborate your answer according to the marks given. State the evidence. Evidence, very important word. अब यहाँ पे evidence from the figure 1.1 that the diagram shows a double circulatory system. Diagram shows a double circulatory system. ठीक है ना? अच्छा, I hope you remember the double circulation. There are two types of circulation in mammals, if you remember. One is called systemic circulation, another one is called pulmonary circulation. Do you remember? I hope you remember. Systemic circulation comes, ये जो heart का diagram है, it's opposite to ours, right side and left side. So ours left side is basically heart's right side. So right side का सारा based on systemic circulation, how blood deoxygenated blood comes from the body parts, entered into the right side of the heart, and then through the pulmonary artery, which is exceptional case, taking the deoxygenated blood towards the lungs, and then how the atrioventricular valves and semilunar valves open, and pulmonary vein takes the oxygenated blood from the carries from the lungs and bring it to the left side of the heart, which is oxygenated blood. So this is how there is one systemic circulation, another one is called pulmonary circulation. So pulmonary circulation, the one which is involved for, I hope you understood, which is involved, associated with lungs. Pulmonary word is associated with lungs. So double circulation ka sara concept is there, because fish mein single circulation hoti hai, you know that. So single circulation mein matlab when blood flows once, so double mein blood flows twice in one circuit. This is a diagram given. This is the graph figure given in most of the books, endorsed books of O levels, IGCSE, latest books, whatever book you are using right now. Figure 1.2 shows the blood pressure of blood flowing through different blood vessels. Jaldi se bring it to your mind the type of blood vessels. You know how main artery ka naam hai aorta. Aorta se jo hai arteries arises, how arteries extend to form arterioles, how arterioles extend to form capillaries, and then capillaries extend to form venules, and the venules veins, and how they form veins. So you can see here कि highest blood pressure जो है aorta पे arteries, you can see here कि arteries पे blood pressure high है, and then it's it drops at the ये X क्या है, ये arterioles, it drops at the finer vessel arterioles, and then capillaries में there is an intermediate pressure. Why intermediate pressure? Because capillaries need time for the permeability towards the nutrients, so that's why pressure is intermediate in between its arteries and vein. So slowest is in the vein. So ye bhi sara jo hai, you must have done that. Blood vessel X, so state the name of the blood vessel represented by X. So aap X ko maine already aapko mention kar diya, what is the blood vessel. Explain reasons for the changes in pressure. You can see here changes in pressure arteries, as already I explained. Using the information figure 1.2, explain again that what is basically that command term here, explain. Explain comes the reason, you have to give the structural adaptation. So bring it to your mind comparison between arteries, veins, and capillaries. So there are two ways of doing the comparison. I always make students work on, if the paragraphs may be comparisons, but I always give them the tabulated forms. Tabulated forms where you understand okay, what are the features on which the basis you are comparing. Suppose I always tell them structural features. So agar he asks you the structural adaptation, so you cannot tell that arteries carries oxygenated blood other than exceptional pulmonary artery. So you have to give the adaptive structural features, size of the lumen, and width of the cell, the parts of the thickness of the collagen and all that. So you can just go through this as well. So very simple. If you have done the comparison, there is nothing very going on with the tough cheese. Uske baad next topic this is whisking, digestion of starch hai. Whisking tube is partial permeable membrane. This type of can come ATP paper also. Dialysis tubing hoti hai, it's a partial permeable artificial membrane, just like cell membrane. So he has given you the he's he's saying there are two test tubes A and B, jo ke placed in water bath at 37, which is at optimum temperature. फिर liquid outside the AB जो है tested. हाँ, there is a term जल्दी से मैं बता रही हूँ, Benedict. Benedict solution जो है, Benedict solution is a reagent which we use for the reducing sugar test. You should know that. For starch, we use iodine as the solution. So यहाँ पे वो Benedict for at 0 minute and after 30 minutes. So ये इसको भी, I think you must have solved it. But if you have not solved it, so you can just pay attention right now. Look at the results. Test tube A में color with the Benedict solution at 0 minute blue है. Original Benedict का color is blue because it consists of copper sulfate, which is blue in color. और color with the Benedict solution at 30 minute में इतने gap के बाद भी still remains blue. It means that there is no action of enzyme. कोई भी अगर वो है starch है तो उसके ऊपर कोई amylase not acted upon and to convert it into reducing sugar, which is maltose or glucose, right? But in B case, initially it was blue in color and then it changed it to red. Bring it to your mind, food test, Benedict's test. It starts from the yellow and then starts from blue and then yellow green in the middle color can come. It depends on the concentration. Higher the concentration of the solution, more it comes in less time, it comes red or orange red color. But if it's dilute solution, that more in between yellow green color can come. So this is what he's talking about here. That's not a it's I think if you have done the Benedict test vagara, so that's not difficult. But I would like to just wait a second, sorry. yes, this is so far. अभी तक we have seen, हमने देखा कि O levels के पेपर में there was no 6 marks question. अभी तक जो अभी specimen O levels का किया, but you can get 5090 wale, you can get also 6 marks question. Now this is a 6 marks question. What is the question? Using the information in the figure 2.1 and table 2.1. So there are two, you have to go back to the figures, figure 2.1 or table 2.1, or explain with reasons for the difference in the results of test tube A and B. Up 6 marks question hai, and you have to write much about it. Your expression should be good. You should have a knowledge of, you should have a knowledge of Benedict test. You should have a knowledge of starch, reducing sugar, so you can do well. Uske baad, this is what that enzyme pepsin, uska substrate kya hai, product or products, product or product. Abhi ek question we just, so question number 1 we did pepsin ka, fatty acid mein usme enzyme kaun sa hota hai, abhi humne kiya hai lipases. So you can trypsin, there are different enzymes. Pepsin is in the stomach or trypsin in the pancreatic juice. और फिर उसके बाद how suppose amylase act upon starch converted into maltose in mouth, and the maltose acted upon by maltase in the pancreatic juice in the duodenum, and how it is converted into glucose. Uske baad state the name of the structure which increase the surface area absorption of small intestine. You must know the villi structure, finger-like projections. This is question number 3. Complete plants. This is based on classification. Bring to your mind plants kingdom. Plant kingdom divided into flowering plants, which are also called angiosperm, which are further divided into monocotyledonous and dicotyledonous. Monocots or dicots, right? And the other simple plants jo hain wo mosses and ferns and bryophytes वगैरह. So ferns also. We did all in you must have done in classification. अब नीचे जो वो पूछ रहा है वो आइडेंटिफाई मोनोकॉट्स. मोनोकॉट्स में सिंगल कॉटिलेडन कहते हैं सीड लीफ को. 1 कॉटिलेडन सिंगल लीफ है. उसके बाद लीव्स आर विद पैरेलल वेनेशन. The vascular bundles are arranged in the stem and randomly all over. They are not uniform, so you can tell a comparison. Uske baad, this is a diagram given life cycle of fern they have given, but you have to focus on ke fern se zyada hamare course mein itna koi life cycle it was not included the whole thing, but he is giving you information during the life cycle. Haploid, ab haploid jo hai means half the number of chromosome, yani like gametes, sex cells. Stages of the life cycle represented by small n. Haploid is always with small n, or jo diploid stage hai wo represented by 2n. For example, in our gametes mein we have sperms and eggs mein we have chromosome number is 23 or 46 is the diploid number 2n. So that's what he's providing you with information, but in a different it's a funds accordingly. So he has given you the he has given you information. Spore, how spore, adult gametophyte is n, iska matlab hai ki it's a haploid. Then how the 2 type of gametes they form zygote as a result of fusion of 2 gametes, and it becomes 2n diploid. And then 2n diploid, just like how embryo jo hai wo develops into fetus, and embryo mein har each and every cell mitotically divide, having a 2n. So this is the same. This is the same as how the mitosis basically. So you have to identify and describe the process, which is also mitosis, meiosis se ye sara kuch relate hai. Uske baad, these are the 2 diagrams he has given you. Shows the parts of 2 flowers of different plants of the same species. You can see here the ab isme kya cheez hai. You have to see whatever the 2 species ke plants are given. Describe what is meant by the term species. So species ki abhi I told you definition. Aram se 2 marks ke accordingly you can write species ke definition. Individuals belong who are morphologically, physiologically, behaviorally they are identical, they have the same structure feature, and they can interbreed to produce fertile offspring. Aapke paas achhi khasi definition hogi. I have given a lot of good notes, and you must be having the good notes. Uske baad pollination ka you must have done self pollination, cross pollination mechanism. Self pollination always favors the homozygosity, same characteristic passing, but cross pollination favors heterozygosity, which leads to variation, which is more good for adaptive features. So annotate. Oh yes, very good term use here. Annotate. I know that most of you are not even familiar with the term annotate. There is one term called label. If they ask you label, you have to just write even single word or 2-word labeling. But if he asks you annotate ka matlab hai brief description of each part. Matlab I can show you that suppose respiratory system in gas exchange. In the diagram, I can write nostrils. I can write the external intercostal muscle ki brief description. trachea ki brief description, bronchus, bronchioles, alveoli. So that is called annotate. ATP paper may be also you will find out annotate. Annotate ka matlab hai brief description, labeling the structure involved, drawing an arrow to show the pathway of pollen. Pollen ka bhi wo, you know that how pollen grain when germinates, so it develops pollen tube having a two nucleus, if you remember, generative nucleus and vegetative. So vegetative withers away. Generative divides into two haploid nucleus, and then one haploid you have fertilizes the fusion nucleus already in the embryo sac to form a triploid nucleus, which leads to endosperm formation. Endosperm nutritive tissue in monocots mostly. Uske baad how the another haploid jo fuses with the egg or ovum in the embryo sac to produce a zygote. So this is called double fertilization. So that's all I've repeated. Explain that disadvantages of cross pollination compared with self. तो आपने अगर cross और self के किए हुए हैं तो they both have their advantages as well as disadvantages. ये कह रहा है कि this is information providing. You know that okay, this is a tree, this is a tree. So he has removed a bark from this tree. Acha, how many of you remember? How many of you transport in mammal? Jaldi se transport in mammal. Source and sink concept. Source is the leaf, or sink is the growing region. All growing region, for example, roots and all, where the food needs to be translocated. Translocation of food in the form of sucrose and amino acid, which are organic solutes, it occurs through the phloem. So there are three evidences regarding phloem meant for the translocation of food. One is called ringing experiment. This type of experiment I did with my students. I always do this. Bark remove ho gaya. This is called ringing experiment. When the bark removed, so it means the phloem they have removed and the xylem. So what happens to the upper part? All the food is going to accumulate here, upper side pe. Yahan pe food is going to accumulate. So it swells here. It will not move downwards because phloem is being removed. So you have to use the information source and sink. So I have given you the source and sink transport in plants chapter. Why leaves in girdle trees are still able to receive mineral ions from the roots? You have to see that. Why glucose made during photosynthesis is required for the absorption of mineral ions by the roots. Glucose is needed for respiration also. Mineral ions are absorbed more through active transport. Active transport needs energy. और वो सारा कुछ बैलेंस इक्वेशन सिंपल बैलेंस इसका मतलब स्टेट द बैलेंस सिंपल इक्वेशन तो सिंपल इक्वेशन का मतलब केमिकल इक्वेशन यू हैव टू राइट तो यू विल गेट मार्क्स व्हेन यू राइट द बैलेंस्ड इक्वेशन केमिकल बैलेंस्ड इक्वेशन उसके बाद आई इज अ सेंस ऑर्गन sense organs are formed from the receptor cells. You know that. Why eye can be described as a sense organ? It consists of basically receptor cells or photoreceptors. For example, there are different types of receptor cells. State two parts of the central nervous system, brain, spinal cord. You must be knowing. This is the eye ka diagram. This is called vertical section of an eye. You must be knowing the parts of the eye, how it starts from the outer, sclerotic sclera, and then choroid, and then retina, and what are the other structure, lens kya hai, muscles hai, ciliary muscles, suspensory ligament, and then all, blind spot kahan pe, fovea, the yellow spot, optic nerve, and all that. So all the parts, ye sare parts given hai, you must be knowing this. This is what that they have given the cornea वगैरह तो ये तो सारा main information है. You need to why a person is able to focus on distant. You know how to far and near accommodation when three how the image is formed. Upside down image formed. You can see the far में देखोगे तो क्या what happens to lens and all that. What happens to ciliary muscles? What happens to suspensory ligament? Lens become thick in near and it become elongated in far and all that accommodation. This is the question. जो कि genetic engineering based है और genetically modified organism. Now it's genetically modified topic है और you can see here कि bacterial cell है तो bacteria में there's a plasmid तो we use plasmid और यहाँ पे plant DNA they have showed instead of showing you the insulin human insulin को भी we can take example. They can give you human pancreatic DNA को remove तो यहाँ पे they have given plant DNA which is it's fine. अगर यहाँ पे human DNA होता तो insulin वाला तो human DNA हो सकता था. You cut the identify the pancreatic insulin gene, then you isolate it, then cut it through the restriction endonuclease enzyme, and then cut the plasmid also portion of it this through the same restriction endonuclease enzyme which we get from bacteria. And you can then insert the cut ends into this one, suppose by DNA ligase enzyme we use, and then you put this recombinant vector, this is recombinant C जो है vector into plasmid free bacteria. Plasmid free bacteria then orders the बैक्टीरिया टू ओनली प्रोड्यूस, जैसे यहाँ पे प्लांट वाला जो प्रोड्यूस करनी है जीन, अगर यहाँ पे ह्यूमन इंसुलिन होता तो यू वर सेइंग ह्यूमन इंसुलिन, बट यहाँ पे प्लांट डीएनए प्रोड्यूस करना है तो इट विल कीप प्रोड्यूसिंग प्लांट डीएनए. उसके बाद राइस प्लांट सेल इन्फेक्टेड विद द जेनेटिकली मॉडिफाइड बैक्टीरिया, राइस प्लांट्स विद द एबिलिटी टू प्रोड्यूस बीटा कैरोटीन. कैरोटीन is important, carotenoid accessory pigments होते हैं which give important for eyesight or vitamin A. So this is the using the information how genetically modified part labeled A in the bacterial cell. A is extracted to cut using enzyme restriction endonuclease forming the ends. ये जो ends आप देख रहे हो ना, there are two types of ends. If you see here ये वाले ends, so these are called sticky ends. So there are two types of end. एक होता है blunt end, we cut from middle से, एक होते हैं sticky end. These are sticky ends. So that the जो exposed bases antigen pair with thiamine, guanine with cytosine, they can be easily जो है joined with one another. This is all. This paragraph is based on basically discuss the disadvantages. Modify, obviously there are so disadvantages का भी और ये बोल कह रहा disadvantages as well. So basically जल्दी से before I end up, this is how that we go about it. This is let me go through this jaldi se. Question number 6 if I start, plasmid or restriction, sticky ends, DNA ligase for joining, recombinant plasmid hota है, recombinant वो जो था the one which produce as result of attached asexual se produce hota है, bacteria binary reproduces rapidly. What are the uses of reproduces rapidly, simple culture demand, no ethical issues, ability to make complex molecules using simple culture solution for bacteria microorganism, additional nutrient content may be too small, seeds may be expensive to purchase, modified organism ka. What are the disadvantages? Unknown health risk may be to consumers, risk of cross contamination with wild rice ho sakta hai, consumers may reject products, और फिर स्कर्वी विटामिन सी से होती है. ये वाला क्वेश्चन नंबर 6 का हो गया. Basically I have discussed with you all the ये this is all based on I वाला है, ये भी very simple, I don't need to ये जो आपके ये इक्वेशन है, ये कार्बन डाइऑक्साइड, वाटर, ग्लूकोज और ऑक्सीजन गिवन आउट, इट्स अ बैलेंस्ड इक्वेशन. This is regarding the photosynthesis. उसके बाद वो जो पूछा था why glucose needed, it's required for aerobic respiration. It releases energy. Energy is needed for active transport. Already told you, active transport is needed for the uptake of absorption of mineral ions from the soil by the root hair. उसके बाद ये translocation process explained you already how what is translocation, और ये sucrose and how the source is the leaf and zinc, all the parts जो हैं they are called growing regions are called it's phloem. Phloem में एक companion cell होता है, metabolically very active, contain mitochondria, sieve tube elements होते हैं. और ये जो girdling cuts the link between the source and sink, so sucrose sugar collect above the girdle. So above the girdle cut करेगा तो वो थोड़ा सा उसमें वो हो जाएगा, वो swell कर जाएगा. उसके बाद ये जो reliant named pollinators, environmental conditions, wind, idea of wastage of pollen, यानी वो कह रहा था ना cross pollination के disadvantage, ये सारे हैं. Idea of more energy needed for cross pollination, more than one plant required, तो ये सारे disadvantages उसके हैं cross के. So cross and wind सबके अपने disadvantage हैं. ये anther है, ये X was ovule, फिर ये label dying, ये जो है I don't think इसमें कोई problem है. Fertilization, meiosis, 600 वो थे कि 1200 थे chromosome number 1200, उसने पूछा था haploid वो 600 बनेगा. Unknown चीज भी information आ सकती है. फिर flowering plant dicot or any two seed leaves, one cot, monocot के features already I explained. So this is all about the enzymes. So this is how to go about it. I have just covered most of okay, I have covered the most of the stuff here, but if anything you need to ask Bukhari, any queries and all that, how to go about it? Okay. Okay, now I have done with all the content. We have so far covered all the exam tips. I have covered all the exam tips. I have covered all the, whatever the needed, whatever needed according to the latest syllabus changes, how 5090 O-level syllabus, according to the new changes, based on all the main topics, whatever the changes, I have covered all the typical questions they can give you. And if you ask me the typical question, what are the important topics, please keep in mind for 5090, whatever the new topics added and whatever the new topics added for 5090, same is the case for the IGCSE. Disease immunity, say you can get question on genetic modification, the questions added, new topics added. So you are going to get more questions on new topics. Genetic material pay you can get on inheritance pay. Yes, ecosystem, impact of man on his ecosystem you can get. Reproduction pay you can get. So that it depends. So I can't restrict. Classification pay you can get question other than whatever genetic material and all. So it depends basically. So any questions or any questions? So do you know anything, any questions?
Thank you so much, ma'am. And I hope this was very helpful to the students. I'm sure it was. बहुत शुक्रिया आपका. You were so generous with your time, and I'm really glad that you were able to help us out here. Inshallah, बहुत सी दुआएं. I'm sure you changed some lives today.
You are welcome.
Ma'am, I can't. Yeah, please go ahead.
It's okay. You are welcome. Thank you so much for giving me a chance again.
All right.
To, communicate with my students.
Thank you. Okay. So should we finish it then?
Yeah, I think because I have done with all. I have covered most of the syllabus changes, everything. I have specimen papers also. I've discussed with with main points and how to go about with the paper, whatever the exam tips, how to time management, everything. I have covered according to my own. I think I have covered each and every aspect of the paper tomorrow. Good luck. I would just like to say
excellent.
Good luck.
Thank you so much, ma'am.
You're welcome. You're welcome.
Good luck, all the students. Thank you. We are all grateful to you for joining in.
Yeah, thank you so much. Ma'am. You will send me the video on. Okay.
Sure, I will.
Allah Hafiz.